We're going to kick it off with Tyler. You're the big face on my screen right now. So plasmid production, right? Plasmids in general get sort of short shrift, I think, in our discussion today, but they are, the quality of them is tantamount, right, to actually getting us to, and our drug substances and our drug products to where we need them to go, right? So arguably, we've made a lot of advancements, right? Through COVID, plasmids are not necessarily a brand new technology, right? Fermentation processes have gone through quite a bit of growth in the past few years. So as you've been surveying the landscape today, what's your take, right, on plasmid production processes? I mean, where are some of the biggest variabilities that you've noted, right, across different vendor platforms? And where are you seeing these variabilities having the biggest impact on plasmid quality today for specifically mRNA development?
Yeah, it's a great question, Anna. And when I think about it, I think it's been a great space for RNA to now come in after AAVs and a lot of other gene therapy companies have really had to rely on plasmids for the past couple of decades. And so I think absolutely the plasmid production process and analytics are much more mature than a lot of the other work that we're going to work on. But I would also say that I think that that critical raw material, being plasmid, is probably the most technologically challenging process when it comes to production, right? You still are reliant on cells. You're going through fermentation, like you said. There's a lot of art in, how you're going to feed those cells, how you push those cells, what selection of strain of cells are you going to actually utilize. So there's a lot of optionality there, which I think can be good, but I think it also can be a lot thrown at anybody that's starting to try to develop their own products. And so you have to really try to start to understand, what does my product need? What are the needs for me as I go from having this plasmid and through linearization into what scale of RNA production and then finally LNP production that you're going to require. But I think the technology is actually is looking really, really promising for plasmids over the last 10 years. Really understanding, I think, the fed batch technology, not having to scale to these large bioreactors just to get better yields, but really focusing on making healthier cells, pushing the density of those, really working on the quality of the plasmid that's going to be produced out of those cells. And a lot of that is being gained because of some of the in-process analytics, where we can get real-time monitoring on, do they need additional glycerol or glucose? What's the lactate levels like? What's the pH looking like? And all of these things can be kind of put together in a way that we can really control the fermentation process. So I think that's been some of the best pieces of the last 10 years working in the plasmid space. And then I think on the downstream side, there's still, a lot of different size plasmids are being produced, a lot of plasmids with different GC content. And so the downstream process, I think, also is something where you need to be looking and understanding what your product requires, what type of anion exchange or Hick chromatography steps do you need to clean up that plasmid to get it to the appropriate quality that you need. And really maintaining good yields through that, right? And so I think there's a lot of options there in terms of chromatography. So I think, there's a lot that has happened to mature that space. Now, with all that being said, it is absolutely the most complex unit operation compared to RNA and LNP. And so I think the one space for us to really question in terms of for RNA and LNP products is, what quality of plasmid do we truly need moving into an IVT? I think there's a lot of questions still there. I think we've taken a lot of what the gene therapy and AAV space needs, and we've just kind of copy and pasted that over. And I'm not sure that's true, right? I don't think we may need the same quality, or there may be different critical quality attributes that we need to be really focused on for good RNA production in the downstream piece. So with that being said, I think there's a lot around, what downstream unit operations are critical, which ones could we forego, and what analytics do we need on the back end to really understand plasmid. So I just threw a lot out there, but that's kind of how I perceive that's how I perceive plasmid right now. And I think the beautiful piece about RNA and IVTs is that we know we're going to really be able to expand on that plasmid and make even more RNA in our IVT, where maybe in the AAV space, the critical material constraint is plasmid. And in IVT, that's not so much the case, but it's always great to have more, right? For
Yeah, absolutely. So yeah, so you've hit on a couple of different things here in terms of, some of the advancements we're seeing in upstream and downstream, but also sort of the lingering questions we have around what really that quality standard needs to be, right? To actually make mRNA as opposed to something like an AAV, right? So, and I think that that remains one of the bigger question marks. And that's been something that for a couple of years, I know we've talked about, right, at different conferences, and how we're going to figure that out, right? That's going to ultimately, a lot of our insights into that are really going to dictate what we're looking for in terms of purity, right? And the
Mhm.
platform for producing a plasmid that we ultimately need, right? So, April, I'd like your take on this topic as well. Are there, Tyler just mentioned a couple of upstream, downstream pieces here that he's looking at and considering and seeing advancements in terms of plasmid production. What are you most excited about, or where do you see some of the biggest process variabilities, right, that are impacting the quality of plasmid today for your mRNA gene editing, right, production?
I agree with everything Tyler said. And I think one of the things I would add is that it also starts with the GMP master cell bank when you're talking about mRNA production with the plasmid. So where we've had challenges and where we've liked approaches with certain mRNA manufacturers is trying to get the poly A tail stabilized. Because if you have an encoded poly A tail in the plasmid, that's great. It saves you a step on your IVT because you don't have to tail as a separate step. However, it's unstable in the plasmid, and you have to really keep an eye on it as it passages. And we've definitely had experiences where if you're working with a manufacturer who really understands how to screen those clones going into your master cell bank, you can make sure that poly A tail length is long enough and then make a big master cell bank so it doesn't keep, because every time you passage, it's going to keep truncating. Yeah, so that's been quite important for us for quality. The other thing I'd mention too is that for me, anytime plasmids for mRNA production comes up, the main question that comes to my head is like, how long are we going to keep using plasmids? Because, past a certain point, I keep looking, I monitor the amplification technology that's been up and coming for a few years now. And, it's not exactly tailored to mRNA production, but I think it'll get there at some point. So to me, like that's also a potential solution is actually not do plasmids.